Welcome to Biligoloji, where we decode the latest breakthroughs in science. What if you could live forever, just by making copies of yourself? It sounds like purest science fiction, but a groundbreaking 20-year study published in Nature Communications just gave us a definitive answer. And it turns out, even immortality has an expiration date.
So let's get right to it. The core question is this: can an animal be cloned forever? Could we create this infinite, perfect line of identical copies, basically preserving a creature for all time? This is one of those concepts that really pushes our understanding of life itself. And for decades, scientists have wondered about it. But now, after one of the longest, most painstaking experiments of its kind, we finally have the answer.
And the answer is this number: 58. Yep, just 58. It might not look like much, but trust me, this single number is the jaw-dropping conclusion to a scientific saga that took more than 20 years to unfold. So, what does it actually mean?
It means that's the hard limit. That's the wall. You can clone a clone and then clone that clone 57 times. But when you try for number 58, the line just collapses. The process completely fails. It is a genetic dead end. It is a biological expiration date stamped right onto the dream of immortality.
So, how on earth did scientists figure this out? How did they land on such a precise, specific number? Well, this wasn't some discovery made over a weekend. No, this is the story of a single marathon experiment that pushed the absolute boundaries of what we thought was possible.
Just try to wrap your head around that for a second. Over 2 decades of continuous, nonstop work. It involved more than 30,000 individual cloning attempts. This wasn't just another experiment. It was a monumental feat of scientific grit and dedication, all to answer that one seemingly simple question.
And the scientist leading this incredible charge is Teruhiko Wakayama of the University of Yamanashi. I mean, this guy is a legend. He has spent his entire career just pushing the absolute limits of cloning. We're talking about cloning mice from cells that were frozen solid for 16 years. He's cloned mice from freeze-dried cells. He even used freeze-dried sperm that had been orbiting the Earth on the International Space Station for 6 years. His ultimate goal? Nothing less than, in his words, permanently preserving the genetic resources of all animals.
This whole journey for him really started way back in 1997. Now here's where the story gets interesting. By 2013, his team had reached 25 successful generations, and they actually published a paper suggesting that maybe this whole process could go on forever. They literally wrote that it may be possible to reclone animals indefinitely, but they kept going, they kept pushing. And as they did, a very, very different reality started to emerge, leading them to this final, definitive limit of 58.
So, we have our limit, 58. But the really big question is, why? Why does it stop there? What's going on? What is this ghost in the machine that brings the whole process to a screeching halt? Well, the answer lies deep, deep inside the genetic code itself.
So to really get this, we first need a quick refresher on how cloning works. The technique they use has this fancy name, somatic cell nuclear transfer. But it's actually pretty straightforward. You take an egg cell, you pop out its nucleus, its genetic control center. Then you take a nucleus from a regular adult body cell, what scientists call a somatic cell, from the animal you want to clone, and you stick it into that empty egg. And just like that, you've got a new embryo that's a genetic copy.
Okay, so here's where the problem creeps in. The best way to think about it is like making a photocopy of a photocopy, right? The first copy you make, it looks fantastic, almost perfect. But then, if you take that copy and copy it, tiny new smudges and errors start to appear. And if you just keep copying the copies, well, eventually the image degrades into an unreadable, blurry mess. The exact same thing happens with DNA. Every single time you create a new clone, tiny new genetic errors, mutations, get introduced. And they just build up and up and up.
And we are not talking about little harmless typos here. As the generations piled up, the researchers started finding absolutely catastrophic mutations. I'm talking about things like the loss of an entire chromosome. That's like ripping a whole chapter out of the instruction manual for life. It's just gone. And eventually, the genetic code simply becomes too broken, too degraded to build a healthy, viable animal.
Okay, so we've got a limit, and we know why it happens. It's a genetic breakdown. But so what? What does this actually mean for us in the real world? Well, the implications of this cloning conundrum are honestly massive.
This quote from Michael Lynch, who's an evolutionary biologist at Arizona State University, just nails the problem perfectly. He said, "Once the mutation is in the lineage, it's there forever. There's no way back. Think about that. With cloning, there is no reset button. There's no undo. Every single genetic mistake gets passed down, creating a one-way street straight towards genetic decay.
And this is in huge contrast to nature's own brilliant solution, sexual reproduction. That whole process is designed to mix genes from two different parents, creating brand new combinations that can hide or sometimes even completely weed out harmful mutations. Cloning, which is a form of asexual reproduction, does the exact opposite. It makes a perfect copy of everything, including every single flaw, and then it locks them in forever.
So why should you really care about this? Because this discovery throws a wrench into some major global efforts. Take agriculture. The whole dream of finding the one perfect cow, the one that produces the most milk, and then just hitting copy-paste forever? Well, that plan now has a time limit. Same goes for breeding a prize-winning racehorse. And maybe most critically for conservation. This shows us that trying to save an endangered species by cloning them isn't some simple, permanent fix. We now have to deal with this inevitable genetic decline.
So after a 20-year experiment, 30,000 attempts, and 58 generations of mice, what's the ultimate lesson here? What can we take away from this absolutely remarkable scientific journey?
I think it all comes down to this. Nature really values change, not perfection, not static copies. Sexual reproduction, with its constant shuffling of the genetic deck, is designed for one thing: adaptation, for resilience. And this experiment proves that even when we use our most advanced, most sophisticated technology to try and force life to stand still, biology's fundamental rules of change, decay, and ultimately, endings, they always win.
And that leaves us with a really profound final thought to chew on. We've always kind of thought of preservation as freezing something in time, right? Keeping it exactly as it is, forever. But if this incredible experiment proves that perfect, static copies are a biological impossibility, then what does it truly mean to preserve a species for the future? Thanks for tuning into Biology. Stay curious, and I'll see you in the next episode.